introduce my co-chair, Dr. Sarah Volkman. She's a celebrated research scientist from Harvard. Her interest is in genomics and transmission. We look forward to your talk, Sarah.
Very kind, Moses. Thank you. Okay, so thank you for the organizers giving me the opportunity to share our work looking at genetic epidemiology of malaria in Senegal. And one of the real challenges for successful elimination of malaria is understanding whether infections in very low burden areas where there are few cases left are the consequences of importation or local transmission. So to address this issue, we applied a genotyping strategy to test whether infections from the pre-elimination setting of Richard Toll, Senegal, represent imported or local infections. So when we look at successful elimination, this will benefit really from identifying the origin and the means of transmission of the few remaining cases in this context. And it's important to determine the risks of importation or vulnerability and the risks of transmission or receptivity in these sort of pre-elimination context. And this type of information can be very useful in optimal targeting and stratification of your interventions to achieve success. So importation of cases can lead to introduced cases in the first generation and then lead to ongoing transmission of indigenous cases year after year. And I'm going to talk about some of these signals in the context of this study. So Senegal is a malaria endemic country with heterogeneous transmission ranging from very low in the north to quite high in the south. And I'm going to be focusing on the setting of Richard Toll here in the northern district, which has a very low incidence, estimated at about 0.2 per thousand in the year 2017. Most of the cases that are detected in this region are found among people who have a travel history, people who've been out of the district for 24 hours or more in the past or previous 14 days. And so we hypothesize that most, if not all, of the cases in this Richard Toll area are due to imported infections. So we applied a strategy looking at genetic relatedness to test for the difference between imported or local infections. And these samples that I'm going to talk about were mostly from passive case detection, along with some reactive, active investigation of households from those index cases. We made pairwise comparisons among the infections within Richard Toll, and we also compared this to another setting of moderate transmission, Chess, Senegal, where we have been sampling for many, many years, looking at an ongoing and long-standing collaboration with Professor Daouda Ndiaye at the University Cheikh Anta Diop. And I'd like to emphasize that this is really just a proof of concept sort of presentation, looking at the relatedness within and between two areas. And the goal is really to gather enough data across all of Senegal to understand how these signals relate in the context of that sort of overall transmission sampling. So we also investigated, as I mentioned, the genetic relatedness within Richard Toll, and I'm going to talk about genetic signals related to year-over-year transmission, which we talk about as persistence, and also look at the relatedness of infections within households. So the study design is really to look at all the passive cases that are detected at the health facilities in this district, and then to deploy a surveillance team that goes out to the household and to neighborhoods, neighboring houses in a reactive active case design. And you'll note that a majority of our cases actually came from the passive cases detected at the clinic, with about 10% only detected among the households sort of as secondary infections. We then took the reactive, the sort of RDTs, the rapid diagnostic tests from this study, and applied a genotyping strategy. So we used a barcode technique that we've talked about many times. It employs 24 independent single nucleotide polymorphisms, or SNPs, that are at high minor allele frequency in the population. And we received 759 samples, of which we were able to get genotyping data for 649 of these. And of that, 473 represented a so-called monogenomic infection, or a single genome infection. So I've given our rules for what is considered a failed. When we miss 5 or more assays, we don't include that data. When we have more than 2 mixed alleles, we talk about this as a polygenomic infection, and the remaining are these monogenomic infections. And we've shown in previous studies that these represent single genome infections, and we also know that the genetic variants in the barcode alleles are sufficient for differentiating infections within Senegal. So we then compared pairwise comparisons to evaluate the relatedness. We scored each of these alleles, whether they matched or there was discordance, to identify barcode groups. And we, for definitional purposes, talk about identical barcodes as those that match at all of the 24 positions. And we talk about highly related parasites as those that are matching out of 23 of 24 of these positions. We then applied these pairwise comparisons, looking at our data set showing Richard Toll samples in green and chest samples in blue. And we were able to separate the unique, meaning those genotypes that only show up once in the population, compared to those that are related or in those barcode groups for these 2 populations. So we looked at the within and between relatedness. And we found a large proportion, 21% of the Richard Toll parasites actually share a barcode signature with a parasite from the chest population. And this is consistent with possible importation. Next, we wanted to look at the genetic relatedness within the Richard Toll population. And for this, just to show you, we took a sampling from a given year, and we identified a genotype, a parasite type, shown in red, and we looked at the appearance of this parasite in subsequent years. And this allows us to look at the year-over-year sort of evidence for that same parasite type or lineage in the population. And we represent these persisting lineages as a sort of stick and ball representation, just to give you a sense of what we're looking at in the next slide, which shows us the data from this actual data set. So here we see evidence for a lot of genetically identical and highly related parasite lineages that are appearing multiple years, year after year, within the Richard Toll district. And although we cannot exclude that some of these may be re-imported at 1 year after the other, many of these lineages are only seen within Richard Toll district samples, suggesting that they are consistent with local transmission. So this year after year persistence of individual parasite types suggests this local transmission. So next we wanted to look at comparing the genetic relatedness within the passive case that was detected at clinic and any positives within the household. And although we had few of these, we were able to score them. So looking at a highly related parasite set shown in red, or if those parasites were discordant, we considered them unique infections. They didn't match. And then we scored these household pairs for travel history. And we were able to see evidence of enrichment, really, for households among those individuals who had no travel history, having genetically identical parasites. These highly related parasites were found, again, enriched in the households of those individuals that had not traveled. And the finding of these genetic related parasites within those households of non-travelers is also consistent with local focal transmission. So our data are really showing us that using genetic evidence, we can differentiate local and imported malaria infections in the pre-elimination setting of Richard Toll, Senegal. We found a large proportion of parasites from Richard Toll are genetically related to parasites from Chess, Senegal, and this is consistent with importation and potentially vulnerability for this population. When we look at evidence for local infections, we find genetically identical parasite lineages that are showing up year after year, again, consistent with this idea of local transmission or receptivity. And finally, when we were looking within the households of those individuals that had a passive case and a reactive case, compared the genetic signatures of those parasites, we found that there was parasite enrichment of parasites that were identical among those households that did not have a travel history. also consistent with local and focal transmission. So we believe that genetic data really provides a high-resolution information that can reveal patterns of parasite relatedness, and that this information can be leveraged to differentiate local from imported infections. And this has the opportunity to guide elimination strategies. So I'd like to acknowledge a large collaborative group that was involved in this project. All of this work is performed in the laboratory of Professor Diane Worth, and I'd like to specifically acknowledge the help of Rachel Daniels, Steve Schaffner, and Dan Hartle with regard to the analysis. All of the sample collection was done with PATH MASEPA collaborators in collaboration with our group, collaborating group from University Cheikh Anta Diop and the PNLP, the National Malaria Control Program in Senegal. And I'd like to acknowledge the gracious support funding from the Bill and Melinda Gates Foundation. Thank you.
Hi, nice talk. Do you have any plan to estimate the genetic differentiation between sites in order to estimate, for example, the gen flow and estimate the migration rates and in which direction?
Yes, excellent question. So looking at genetic signatures and ideally going beyond 24 SNPs, looking at whole genome sequence to look at the relatedness, the identity by descent, we should be able to track the relatedness among and looking at the similarities and differences among populations to estimate gene flow. And we think this is a really important way of understanding what are the intervention units and where we're going to need to target interventions. So thank you for that question.
We have some time for additional questions, if any. Well, if there are no questions, thank you very much.
Thank you.